So today I have the pleasure of talking to Professor Thomas McDade. He's a professor of biological anthropology, which I think is just awesome, by the way.
Thank you.
Yeah, he's also a faculty fellow at the Institute for Policy Research and director of the Laboratory for Human Biology Research. Welcome, Professor.
Thank you. It's a pleasure to be here.
So if you don't mind, just to get us going, can you give us a quick summary of your journey through science? So where did it all begin for you?
Yeah, that's a great question. So I was always good at school, and I was always good at math and science. And when I went to college, I just assumed that I was going to be pre-med because I was interested in health and medicine. And so I took all the pre-med classes, but they're a grind, as you may know, and many of your listeners will know. And I started to realize that I was more interested in research and less interested in the practice of medicine, probably. And I had the great fortune of connecting with an anthropologist at Pomona College, where I went to college, who introduced me to a different way of thinking about health and human biology, and that it was an biological anthropological perspective. So he really mentored me in the sense that he showed me different ways to think about the things I was interested in. So then I decided to go to graduate school in anthropology with an emphasis in biological anthropology, and finished my degree and got a job at Northwestern a year later, and I've been here ever since. I just finished my 20th year, and it's been a great journey. Northwestern's been a great place to be. We have a terrific anthropology department, and it's been a great place for me to do my work.
Awesome, awesome. So biological anthropology. So for me, anthropology is one of those vocations that just oozes with romance, right? So what is biological anthropology in a nutshell, really?
Yeah, well, anthropology literally means the study of humankind in its all forms, its history, its culture, society. Biological anthropology has to do with things that have to do with our evolutionary history, our relationship to other species, and the particular area of biological anthropology I'm engaged in is called human biology. And we study living people, but we tend not to study them in a clinical setting. We tend not to study people who are sick. We tend to study everyday people living their everyday lives all around the world to try to understand how is it that the factors that we live in, how we live, where we live, who we live with, the conditions that we grew up in, how those shape our physiologies, and ultimately how they determine our health. And it turns out that, a clinical perspective on medicine and health is very useful, obviously, but it's based for the most part on the study of people in the United States and other rich countries around the world, and a lot of it's just based on lab rats, literally. So if you want to take a more comprehensive understanding of human biology, you need to take a broader perspective and consider a broader range of places that people live their lives, and that's what we do in biological anthropology.
Interesting, interesting. Well, hopefully one of these days I can take an anthropology course and, maybe you'll be one of my professors.
Yeah, I hope so. No, great. We can talk about that later.
All right. So I found out about your work listening to the Breakthroughs podcast by Weinberg School of Medicine, and they had Dr. Elizabeth McNally on, and your work really piqued my interest. So I'm interested to know, how did that relationship materialize? How did you guys come to work together?
Yeah, it's an interesting story. I've never met Dr. McNally. I still haven't met her in person, actually.
Really?
Um,
Wow.
yeah, we became connected post-COVID lockdown, and we got connected through a mutual colleague, a colleague and friend of mine. And I was saying to him early on, this was in April after the lockdown, that I was interested in developing a method for measuring coronavirus antibodies. And I wanted to do that in dried blood spot samples, which are drops of blood collected on filter paper and dried on the filter paper. I've been using that method in my lab at Northwestern and in my research for more than 25 years, because for about $1 and for very little inconvenience on the part of the research participant, I can get blood from anyone, anywhere around the world. So it's a really easy way to get blood in a remote field-based setting, outside the lab, outside the clinic, and get the blood back into the lab, where we can do analyses for various things. And that's been fueling my research for 20 some years. I realized in the current moment of the pandemic that antibody screening was probably going to be important. We've developed many antibody assays in my lab before, so we had the expertise for doing that. But the other problem was that everyone's being told to stay home. And, you can't get blood from people in their home if you're doing it the traditional way with a venipuncture blood collection.
Okay.
So this is why I thought, well, the blood spot sampling procedure is going to be an interesting way to go. We can send collection kits to people in their homes. They can send the samples back into the lab, and then we can analyze them. So I was talking to this colleague about this, and he said, oh, well, there's a colleague of mine at the medical school, Dr. McNally, who is working up an antibody assay for clinical purposes to screen healthcare workers. And they're going to draw venous blood. And so she and I got connected, and our groups got connected, and we developed these protocols in parallel. She developed a serum protocol. I developed a blood spot protocol. We're using basically the same supplies. And we've had several Zoom calls, lots of conversations, but I still haven't met her and the rest of her team in person.
Wow, interesting. I mean, it's a sign of the times, right? When we can do, groundbreaking research without actually seeing eye to eye, right?
Yeah, that's right. That's right.
So before we had this interview, you sent me a couple of your papers, and I want us to talk about your work in the Philippines, right? And if we can start off with this idea that socioeconomic status can actually have epigenetic consequences. Now, this is as fascinating as it is troubling, right? So can we kind of dwell on that for a bit?
Sure. So let me take just one step back and just so all your listeners are aware of what we mean by the term epigenetics. We all inherit, we get genes that we inherit from our parents. Those are fixed at conception, fixed at birth. But epigenetics refers to a series of biochemical processes that change the structure of the genome and the access points for transcription factors. So it's one thing to have a gene, and you can have a gene that does horrible things, but if that gene is never expressed, it doesn't matter if you have that gene.
It doesn't matter if you have it, yeah.
And so these epigenetic modifications turn some genes up, turn some genes down. It's like a dimmer switch.
So DNA modification and histone methylation, right?
Exactly. There are various epigenetic processes that lead to this process of turning up and down genes in terms of their regulation. And it turns out that for a lot of genes and for a lot of locations on the genome, your experiences leave a mark on the genome that subsequently affects future expression. So we were interested in this study in the Philippines where the participants were literally born into the study in the early 1980s, and they've been followed up multiple times since. And in 2005, we had an opportunity to collect blood samples. They were very willing to give us blood samples, and we could measure patterns of DNA methylation, which is an important epigenetic process. And we looked at that in relation to early life socioeconomic adversity. And our idea was, or the hypothesis was, that we know that people who are born into poverty are at much higher risk of dying earlier or later in their lives and having more health problems over the course of their lives. That's an established epidemiological fact. How does that happen? And it's important to understand the underlying biology if we want to intervene in some way. And so we hypothesized that epigenetic modifications might be part of that process. And we did find that individuals who were born into poverty had significantly different methylation profiles on a large proportion of their genome, almost 10%. So to me, that was strong evidence for the impact that socioeconomic context can have on the structure and function of our genes. Now, we don't really know exactly what that means for health in the long term in our study, but we're going to follow people up and we're going to find that out.
All right. So I can understand how socioeconomic status can have physiological ramifications, right? But the fact that, it could have epigenetic ramifications, that's a bit scary, right? So are we saying some of these ailments, some of these pathologies could actually go down the germline, right? And basically affect the progeny of these subjects?
Well, I think there are two important points there. One is there are a lot of people interested in that very question, the fact that epigenetic modifications in one generation could be passed down to the next generation. There's actually no direct biological evidence for that happening in humans at this point in time. There's some evidence in animal models, mice in particular, that can happen. And a lot of people are trying to find that out in humans, and no one has yet found that out. So we just don't know if that's true or not. The other thing, so it might be, and if it is true, that would have tremendous implications, as you're suggesting. But it's also important to keep in mind that just because something has an impact on your genome or your epigenome, it doesn't mean that is fixed.
Okay.
the different experiences later in life can undo some things in your epigenome and do some things at different parts of the genome. So just because you had an adverse experience and it might have had some impact on methylation, that doesn't necessarily mean that's fixed for the rest of your life. So you don't want to be overly deterministic about it.
Deterministic about it. Okay. So with that being said, it just kind of worries me about what we're going through now with the pandemic, right? So the fact that certain demographics in the country seem to be feeling the ramifications a bit more than others, and
Mhm.
it almost rings true to the work you're doing and what you're saying. So we do need to look at our we do need to cross check how we actually carry out medicine in general and how we take care of our citizens, because it seems like some people are being left behind and it's having just tremendous health effects, and I think this is something we're going to have to deal with for a long time, right?
Yeah, no, I totally agree. I was just looking at the numbers in Chicago yesterday. Latinos are 4 times as likely as whites in the city to become infected with COVID-19. African Americans are 3 times more likely to die of COVID-19 than whites are, and they're more likely to die than Latinos, even though the Latinos are more likely to get infected. So you can't explain that with underlying genetics. There's no basis for that. It has to do with social conditions, physical conditions in different environments, and different developmental histories of these groups of people. And this is where my training as a biological anthropologist, I think, comes into play. I tend to think of these problems or these kinds of health conditions as products of development and products of social relationships rather than just as individual level behaviors and clinical syndromes. And you think about how everyone's talking about COVID as a disease. Our understanding of that disease is based almost exclusively on the study of the sickest people in hospitals.
Sure.
But think about how the disease is spread. It's a community problem. That is where the virus is transmitted, and that's where the virus is having its most dramatic social and economic impacts. So really, if we want to understand what's going on and prevent future outbreaks and prevent the adverse impacts on some demographic groups in our society, we need to understand what's going on in the community. And that's been motivating my research to develop this minimally invasive approach to testing for antibody screening, because we can identify where the virus has been, what groups of people are most likely to get infected, and what factors promote the distribution, the spread of the virus in the community.
Interesting, interesting. So just a few days ago, I was talking to another professor that actually does dynamic modeling when it comes to COVID-19, and something just hit me. It almost seems like socioeconomics is almost like you know, a comorbidity of this disease in a way.
Mm-hmm.
Right? So it goes beyond just physiological comorbidities. Even poverty is, to me, a comorbidity at this point.
Absolutely. So poverty is a risk factor for just about everything. Unfortunately, it makes your body more vulnerable, makes your social structures, the resources you have, you're just in general more vulnerable. And the COVID moment is exposing this. And actually, I think raising awareness for a lot of people in the United States about the problem and the contribution of poverty to it. And I think a lot of people don't really realize that. And we tend to think of health as a product of individual behaviors and decisions. And therefore, if someone gets sick, we could sort of blame them for having bad genes or deciding to smoke or not exercising enough. When in fact, it's really the broader social structural factors that may, in some cases, limit your access to good food and ability to exercise safely and your ability to access good healthcare. So really, you can think of health as a product of more social phenomenon and context. And the COVID epidemic is a dramatic demonstration of that.
That's right. So let's segue into why I wanted this discussion to begin with, right? So testing for COVID-19 has been the cause célèbre, in the country and around the world, right? So why are the tests we currently have so prone to false negatives, right?
Mhm.
So these, RNA, dependent RNA, polymerase, chain reaction tests, why are they so prone to false negatives?
Yeah, so I mean, it's important to keep in mind there are 2 types of tests. One is the PCR test, like you're saying. So that's a direct test for the presence of the virus.
Yeah.
Uses polymerase chain reaction, PCR, to amplify the genetic material of the virus and detect it in a sort of a yes/no qualitative kind of way. The reason that there are a lot of false negatives, I mean, there are probably some technical issues as well, but it really has to do more with the biology of the virus. when you have that swab stuck up in your nose, and people say it's like, feeling like it's scratching your brain,
So the nasopharyngeal swab, right?
the nasopharyngeal swabs, the virus has to be present in the tissues that are being sampled at that moment. But virus expression can be transient. Or you could have a lot of viral expression at the early stages of the infection. Then you finally get into the doctor and you finally get tested. The virus has stopped expressing itself. Therefore, it's not detectable, but you're still suffering from the symptoms due to your inflammatory and immune response to the virus. So in that case, you'd be diagnosed negative based on that test, but you clearly had it. And if you'd been tested maybe 3 or 4 days before, then you probably would have tested positive. So that's why there are a lot of false negatives with that test.
Interesting. All right. So does the lateral flow immunoassay test seem to solve some of these problems we have with our
Right.
regime?
Well, so then there's the PCR testing for the virus, and then there's antibody testing, which tests for the presence of antibodies, which indicate your body's immune response to a prior infection. So antibody testing is not very useful clinically. So your doctor is not going to order an antibody test to help determine in that moment, is your fever due to COVID or is it due to influenza, right? But after the fact, it can tell us with a high degree of accuracy whether you have been exposed. And this is important because a lot of people are exposed and actually don't develop symptoms or have very mild cases. And so they don't get registered as cases of COVID-19. So it's important information for us to know just where the virus has been and how many people have been infected. Okay. The best tests use serum or plasma, which is collected from venous blood, so venipuncture, the classic way of getting blood that everyone's familiar with. That allows you to take the blood into the lab in a hospital or another laboratory facility and test it using very rigorous and accurate methods. Another approach, the approach that was rolled out early on when antibody testing started to come online, are the lateral flow test kits. And these are little cartridges that where you take a drop of blood, it can be from your arm, it could be from your finger, and you put it in the cassette, and the blood moves down a membrane. And if you test positive, this colored line emerges. And it's the same principle as a pregnancy test. And those are very useful because you can use them in people's homes. You could take them into the community. There was a study that did it in parking lots in New York and the Bay Area. But the problem is they're not as accurate and they're only qualitative. They give you a yes, no, and they're prone to false negatives and false positives, actually. And a lot of the early tests were rushed to market, and they weren't properly vetted, and they were very inaccurate.
Inaccurate. All right. So this brings us to dried blood spot sampling, right? The work you're doing right now. So the blood is collected on a filter paper, then it dries up, then it's shipped to the lab, right? So right. Do we envision test kits being sent to people's homes and maybe job sites? Is that the
So the idea is that this approach sort of combines the convenience and safety of blood collection in the home. So you're not having people come out of their homes, and you're not having them go to a hospital, which is already overwhelmed. So they can collect safely and conveniently in their home, but they send the sample to the lab, or we use the same procedures with the same level of accuracy as the gold standard serum-based protocol. So it combines the best of both. And the idea is, that we can use this as a research tool or a public health screening tool to identify people of interest and send collection kits to them, or bring collection kits to their job sites, or to a community center or a church, have people collect the blood themselves and then send it back into the lab. We just launched a study called SCAN, Screening for Coronavirus Antibodies in Neighborhoods across Chicago. And we've selected 10 neighborhoods in Chicago, and the neighborhoods represent 5 pairs of neighborhoods, adjacent neighborhoods. One neighborhood has a higher than average COVID caseload. The other adjacent neighborhood has a lower than average COVID caseload. And we're recruiting up to 3,000 people total, hopefully, across those neighborhoods. And we want to see where is the virus in those neighborhoods. Is the caseload of COVID-19 higher because there's more transmission of the virus there? Or is the level of the virus about the same, but other predisposing factors are leading to more infections and more mortality in some neighborhoods. And we have a survey, a web-based survey that's part of the protocol as well. So people provide some information on their behaviors. Are they wearing masks? How many people are they living with? Are they designated as an essential worker and therefore don't have the choice to work from home the way I do? And that might be putting them and their families at risk. So we'll be able to determine some of the community level as well as individual level factors that predict exposure to the virus.
Interesting, interesting. So I'm sure people bring up the issue of safety, right? So are these packages safe to be shipped in the mail? So what sort of safety mechanisms are put in
Yeah.
place, or is this just an intrinsic, I guess, property of this process?
Yeah, no, it's a great question. We all should be concerned about safety, especially right now. So the papers, when you collect the blood on the paper, it disperses and it dries the blood. And dried blood, when blood dries, it neutralizes virtually all pathogens in the blood. So we still need to be careful with it and treat it as if it is infectious. But in comparison with liquid blood, there's no way to, it doesn't get spilled. There's nothing to get broken. And if you put it inside a plastic bag and then put that plastic bag inside another envelope and seal it all up, it's totally safe for mail carriers and anyone else who might come across the sample. And in fact, the papers were originally developed in the 1960s for neonatal screening programs. So every baby that's born in a hospital in the US has its heel pricked, and then 5 drops of blood are put on these papers. And those papers are collected in hospitals, and then they're sent to a central testing facility. And each state runs their own neonatal screening program. So the cards were originally designed to collect blood safely, small volumes from babies for whom it's hard to collect venous blood, obviously, and then to send them to a central processing facility. So they've been routinely used for 50 years for this kind of application. They just don't get used very much for research applications. So it's me and a handful of colleagues in anthropology and epidemiology have been using this to advance our own research.
Wow, interesting, interesting. So going through your papers, something that really stood out to me was this idea that you basically dwelled on, the fact that you see testing transcending, this binary positive or negative, test results, but it could really open our eyes to other factors, right? So socioeconomic factors, racial factors, apparently epigenetic factors, right? So can we talk about that? Like, because to me that just goes beyond, just the science of, doing your work. This is actually something very, very important. And I think we all should actually see our work in that light.
Yeah. I mean, for me, that is one of the main things that motivates me and drives me to do what I do and how I do it. So much of what we understand about human health and human biology is based on the study of relatively privileged people who have access to high quality healthcare. And then a lot of it, as I said before, is based on the study of lab rats. So we don't really have a good sense of what are the factors that promote health and also put some people at risk in the community where people live their everyday lives. If you want to understand how social factors, how community level factors, how exposures to violence and discrimination, how poor quality diets, things like that, how those affect people's health, you have to go to where the people are living their lives, and you have to go to their communities. You have to talk to them, and you have to hear what they have to say, and you have to work with them and collect biological data from them and with them. And only through that approach, I think, can we shed light on the inequities that affect some racial ethnic groups in our society and people of lower levels of education, socioeconomic status. Because those people aren't represented often in the kinds of studies that generate our knowledge and understanding of health and disease.
Oh, thank you. Thank you. I really appreciate that. So to wrap it up, let's talk about ELISA, right? So this would be the enzyme linked immunosorbent assay. Now, is that the most accurate antibody test we presently
Yeah,
have?
yeah, that's the gold standard approach to quantifying antibodies. It applies to a lot of different things, but yeah, it's a technology where you take a plate, a 96 well plate, plastic with 96 wells in it, and you put some antigen. In our case, we put a little piece of the virus. It's not live virus, but just a little piece of it. We bind that to the bottom of the plate, and we put in our sample. And if you have antibodies against the virus, your antibodies will then stick to the virus, which will be stuck to the bottom of the plate. then we wash everything, and then we add a solution that generates a color change that's directly proportional to the amount of antibodies that you have in your sample that are now bound to the bottom of the plate. And we can put it in an instrument which very precisely can read the amount of color change. And we have samples of known concentrations of antibodies, and we generate what's called a standard curve. And we compare a sample with an unknown quantity of antibody against our standard curve to figure out just how much antibody is in that sample. It's been used since the 60s. It's well established, widely used quantification technique for biological samples.
Wow, I really appreciate the fact that, science that we've done, several years ago, even before I was, even before my parents were born, is, they're basically the champion and saving the day at this point.
Well, we'll do what we can, but you're making me feel old by talking about how your parents weren't even born then. They're probably my age.
Actually, I think my mom was born in the 60s.
Yeah.
My dad too. Yeah, but all right, that's
all right. Well, I'm not quite that
old. Point. That's the point. So, Professor, is there anything else you want to touch on? Anything we didn't?
I don't think so. I appreciate you getting the word out. I think there's scientists, we do our work, and we often don't do, we spend a lot of time talking to each other and our students. We don't do a very good job about reaching out to the public, but in the current moment, as you said, and we were talking about earlier, it's all the more critical to do that, to inform people about what we're doing, and for us as scientists to make that effort and make what we do relevant and understandable by the general public. So I appreciate you taking the time to talk to me and get the word out there as well. It's an important service.
And I do thank you for actually taking the time out to, elucidate these issues and, talk to us about your work and make it digestible for us, because at the end of the day, that's what matters, right?
Yeah, I appreciate that. Thanks very much.
So,
it's a pleasure.
Yeah, so if anyone wants to contact you, maybe undergrads, graduate students, they want to, get under your wing and, toe the line.
Send me an email.
Sounds good.
I'm easy to find.
All right. Well, in that case, thank you, Professor McDade.
Okay.
It was awesome.
Pleasure. All right. Stay safe.
You too. Bye.